Hello, everybody, and you're very welcome on behalf of the Irish Longitudinal Study, TILDA, and the School of Medicine at Trinity College Dublin. We're absolutely delighted and privileged to host our colleague and I think friend, Professor Steve Horvath, who is Professor of Human Genetics and Biostatistics at UCLA Fielding School of Public Health. Professor Horvath is an internationally renowned aging researcher. Not he personally aging, but his work is very much focused on this field and lies at the intersection of several fields, including epigenetic biomarkers of aging, genomics, genetics, epidemiology, aging biomarkers for clinical trials, and comparative biology. His contributions to the study of aging are seminal and extend across many different fields of investigation, and his credits include the development of a widely used systems biology data analytical approach known as weighted correlation network analysis, the development of the first epigenetic clock for saliva in 2011, the development of the multi-tissue epigenetic clock in 2013, which bears his name, the Horvath clock, and I'm quite sure everybody online is very familiar with that now, and more recently, the development of a pan-mammalian epigenetic clock, some of which data he will present to us today, and we're very excited to hear that new data, Steve. His work in the field of epigenetics has been nothing short of revolutionary. To date, he has accrued more than 75,000 citations and has a H-index of 121. He's already received several awards for his work on the genetic clock, including the 2017 Allen Distinguished Investigator Award for Clock Studies in Vertebrates, the 2019 Open Philanthropy Project Award for Mechanistic Studies of the Epigenetic Clock, and the same year, the Schober Award for Outstanding and Innovative Research in the Field of Aging. He holds a BSc in Mathematics and Physics from the Technical University of Berlin, a PhD in Mathematics from the University of North Carolina at Chapel Hill, and a Doctorate of Science in Biostatistics from Harvard School of Public Health. We are absolutely delighted to welcome you, Steve. And as I said, the last time I had the privilege of pleasure of meeting Steve over dinner in Rome, we agreed that he would come to Dublin and give a lecture. And of course, COVID broke shortly after that following February, but we will meet that promise, Steve, in the near future. However, in the interim, we're absolutely delighted that you're able to be with us today, share your most recent research, and I would encourage those of you listening to please put your question into the question box. We'll collate the questions, and Cathal McCrory, who has worked very closely with Steve over the last number of years, capitalizing on the TILDA data set and Steve's wonderful work, will actually curate that particular part of the meeting. So I'm delighted now to hand the floor over to you, Steve. Thank you.
Oh, yeah. Thanks you so much for the very generous words and introduction. It's a real privilege to be invited to you. The TILDA cohort has done very important studies for epigenetic clocks. I will briefly touch on it. So really wonderful to talk to you today. I will talk about methylation studies really in different mammals, but there will be quite a lot of material in humans as well. I will start with some basic facts about cytosine methylation. So remember, cytosines is one of the nucleotides in the DNA. And cytosine methylation allows you to estimate chronologic age and mortality risk. And I use that to introduce a nomenclature about different generations of epigenetic clocks. So the pan-tissue clock was really developed as an estimate of chronological age based on cytosine methylation in many different tissues. And so I call this kind of a clock, an age estimator, a first generation clock. And then several years later, several people developed mortality risk estimators. So we use methylation to predict morbidity, mortality. And this is often referred to as a second generation clock. So for example, Grimm age, it would be a second generation clock. And more recently, we are working on so-called third generation clocks that are defined as multi-species age estimators. In other words, you could have blood samples from different types of primate species, humans, chimpanzees, and so on. And you want to build one regression model or one machine learning model to estimate age in these different species. So that's a third generation clock. And so why do that? Well, these clocks are already being used in human clinical trials of anti-aging interventions, but also we are, of course, very interested in applying such clocks in preclinical studies. So, for example, in mice and rats, and of course, dogs or many other models. And I want to go back in time. So in 2013, we already knew that the pan tissue clock applies to chimpanzees. And this is figure 4 in the article from 2013. And to me, this was a stunning figure. So you see a in the left panel shows you bonobos and the common chimpanzees, and the correlation was 0.9. So it was amazing that this human clock applied to another species. However, in the same figure 4 of the paper, we already find that the clock actually does not apply to gorillas. So although the correlation is like 0.51, but clearly it doesn't work. And this made sense because when you look at the evolution of primates, you will find in the right-hand side that humans are more closely related to chimpanzees than to gorillas. There's an evolutionary distance. So when you just look at that evolutionary tree, you will see there are certain primate species, such as lemurs in Madagascar, that separated from our species about 60 million years ago. And to remind everyone, the dinosaur extinction happened around 66 million years ago, but shortly thereafter, there were already the precursors of primates. And anyways, primates separated about 60 million years ago, very distinct species. And the question is, well, can we ever hope to build an epigenetic clock or a multivariate regression model that estimates age in all of these different primate species based on cytosine methylation? And how do I define it? In highly conserved regions, right? How would you do such a project? You would identify stretches of DNA that are highly conserved across primates, measure methylation, and fit a regression model to build such a clock. It's a risky project, but it works. So I just put a paper in bioRxiv that describes a pan-primate clock. And in that study, we used data from 37 different primate species. The panel A here shows how accurate that clock is. The dots color different primate species. For example, this pink color, these are human samples, but the different panels show you different primate species. And panel I shows you all the lemurs. We had many different lemur species combined. In the middle, in panel E, you see how this pan-primate clock applies to humans. And you see the correlation is 0.98, and the median error is like 2 years. What does that mean? This pan-primate clock is as accurate as our clock from 2013. It's same accuracy. However, it now applies to all of these different primates. And going back in time, there was another finding in the paper from 2013, which was that induced pluripotent stem cells are perfectly young. This was a very robust finding because I looked at many data sets, and what these data showed is if you start with primary cells from adults and you then apply these Yamanaka factors to create iPS cells, the resulting age estimates were negative. Negative age means prenatal age. But then you can ask the same question. Well, does this translate to iPS cells from non-human species? Do they also have a perfectly young age? And the answer is yes. So Vadim Gladyshev, for example, showed back in 2017 that yes, iPS cells from mice also have that property. But you can ask, what about other species? For example, naked mole rat, very different species. And here I show you the same finding. So we have a clock that applies to naked mole rats. And again, we find that iPS cells from that very distant species also exhibit very young ages. Now, we know now that in 3 species, iPS cells are perfectly young, you know, but the question is, what about, is that universal? And I don't have an answer to that question yet. But why do I mention iPS cells? Because it gives rise to an idea. In order to create iPS cells based on Yamanaka factors, you typically have to wait a while. I want to say 30 days or so before the transformation is achieved. And so several groups have had the idea of applying the Yamanaka factors only briefly, in a transient way or pulsing it. And there's quite some literature that this actually rejuvenates the epigenetic clock, or more generally biologic age. It has beneficial effects without changing the cell's state, or the cell type, I should say, you know. And so there are now several groups and even companies that explore that aspect, transiently apply Yamanaka factors or equivalents to rejuvenate us. I want to now come to the mammalian methylation array that I use in all of our mammalian studies. I should tell you right away, it's an infinium array, very similar to the human infinium arrays that you can order, like this 450K array or the EPIC array. So it's the same technology, but it applies to mammals. And you can get access to it through a non-profit, which is known as the Epigenetic Clock Development Foundation. So yeah, the array is widely available, and it profiles 37,000 highly conserved CPGs. And I just want to give you a glimpse of the logic. So here you see nucleotides in different species. The upper row shows you a nucleotide sequence in humans, and it of course ends with CG, the CPG that we want to measure. But this particular sequence is highly conserved in rats and even in bats and so on. But occasionally there are mutations. So the idea of the mammalian array is to use probes that are robust to these mutations. And therefore, we can measure cytosine methylation in these highly conserved stretches of DNA. And here I show you our pan tissue clock for the naked mole rat. I highlight this particular species because many people believe it has negligible senescence. Certainly on a phenotypic level, this animal does not seem to age. It has a remarkably long maximum lifespan of 37 years. However, if we carefully look at it, we can see that according to epigenetic clocks, the naked mole rat actually ages. And we had tissue samples from young animals from age 0 to 5 and up to 10. And you even see, so the x-axis shows you chronologic age. And you can appreciate that even when these animals are younger than 10 years old, there's already a fairly strong correlation with age. And so yeah, on a molecular level, that animal actually ages. And the tissues were provided by Chris Fawkes and Vera Gorbunova and Andrei Selyanov. I now want to come to multi-species clocks, the so-called third generation clocks. So here is a study of elephants, and there are two types of elephants, the African elephant and the Asian elephant. And there, of course, I often like to include humans. And the left panel shows you results for all three species. The red dots are humans, the green and the purple dots correspond to blood samples from these two types of elephant species. And what do we see? Correlation 0.97, which is remarkable. But I want to come to a technical point. If you look at the x-axis here, you will see a number between 0 and 1. Why? Because this is the so-called relative age measure. So what we do is we divide chronologic age at the sample collection by the maximum age of the species. So the maximum age of humans is 122. In other words, a 61-year-old person would have a relative age of 61 divided by 122, which equals 0.5, you know. So relative age measures are a very crude way to align the ages of different species. And I will use this trick throughout my presentation. So we use relative age as the dependent variable in a regression model and regress then relative age on cytosine methylation levels. And that's our regression model. So that's how we build estimators of relative age, which apply then to many species. We have a consortium, which is called the Mammalian Methylation Consortium. We are over 160 investigators. We have profiled over 260 species, mammalian species to date. And I want to show some results. Here I show you a so-called universal pan-mammalian clock. As a matter of fact, these are two different clocks. As you can see from the labels on the x-axis, there's a clock number 2, and clock number 3. Each dot is labeled by a different species, and you observe correlations 0.98, you know, so absolutely, or certainly above 0.9, you know, which is amazing because we have very distant species, including marsupials. So the middle panel shows you how the pan-mammalian clock performs in marsupials. And the correlations are still very accurate, even though far fewer cytosine on the array apply to marsupials, you know. And yeah, so what's the difference between these two clocks? The first clock, again, as you can see from the lower panels, actually estimates the relative age of these different species. If you carefully look at the x-axis, this is again a number between 0 and 1. So this clock number 2, the pan-mammalian clock, estimates relative age. But there's a problem with relative age, which is the following. For many species, the maximum lifespan is actually not known, you know, or really can only be estimated very approximately. But there's a workaround, which is, well, don't use maximum age to calibrate these different species. Rather use a species characteristic that can be more readily assessed, and that would be age at sexual maturity, you know. So our clock version 3 is again a pan-mammalian clock, but it standardizes ages based on the age at sexual maturity of the species. Now, to give you a sense about how well these pan-mammalian clocks apply to humans, I show you in the upper left the application to human tissues, and you see a correlation around 0.95. So these clocks clearly work quite well in humans. Now, in mice, the upper middle panel, the correlation is again 0.95. So yes, it works fortunately in mice. The other panels show you accuracy in dolphins, in dogs, in elephants, in bats, you know. So these pan-mammalian clocks work very well, even if you apply them to a specific species. I want to briefly digress. While most of the talk today is all about estimating chronologic age, I want to briefly mention that you can use cytosine methylation to accurately estimate the maximum lifespan of a species. So here in this plot, each dot corresponds to a different mammalian species. And on the x-axis, we have the maximum lifespan log transformed, and on the y-axis, we have the predicted value. And by the way, needless to say, this is based on cross-validation, you know. I should make this technical point because I show you very high correlations, and the statisticians among you probably think, oh, these are all training sets correlations, but it's not. Every correlation I show you is based on cross-validation or test data. It's always unbiased. In any event, you see a very high correlation. So messages, you can estimate species characteristics quite well based on cytosine methylation. I now want to come to a curious clock. This was a collaboration with Tim Hoare. We developed a dual species clock that applies to humans and sheep. And same trick, relative age between these two species. And so why human and sheep? Well, in sheep, we wanted to evaluate the effect of castration. And it turns out that castrated sheep, male sheep, actually have ears that are younger. And you see this here on the y-axis. I show you epigenetic age acceleration in these ear tissue samples from these castrated rams. And the blue dots tell you that, yes, the castrated males had younger ears. And I can read your thoughts. You probably think, I also want to get younger ears and want to line up for castration, but I need to tell you, I also evaluated castration in blood samples from horses, from cats, from dogs, and it never had a beneficial effect according to epigenetic clocks. So castration rejuvenates probably some of your cell types, or that's the wrong word. It means slows aging in some of your cells, for example, skin cells, but not in other cell types such as blood. I want to also mention we have clocks that apply to all dog breeds. So we looked at 93 different dog breeds, and we have again, we have here a dual species clock, humans and dogs. So you can give us a blood sample or a saliva sample from yourself and your dog, and we can measure the age very accurately. I want to also mention pigs. Why? Because mini pigs are a very attractive animal model in preclinical research. So we have these human pig clocks, that correlation 0.93. I mean, in pigs, very accurate. I mentioned the human cat clocks from Ken Raj and Balázs Sladovec. So I think we have really arrived at a new era of testing. We can apply the same epigenetic clock, meaning the same mathematical formula, the same cytosines, to several species at the same time, primates and so on. And that really allows us to increase the odds that research findings in animal models will actually translate to humans. Because imagine you have an anti-aging intervention that rejuvenates many species according to a multi-species clock. Chances are it will apply to humans as well. I now want to come to a very important question, which is cytosine methylation causal? And by the way, I don't claim that we have the answer. It's really a yes and a no. One can find plenty of examples to support both points of the view. And for example, evidence in favor of yes, that cytosine methylation is important. There are nice studies in the literature. David Sinclair, for example, showed that TET enzymes are needed for optical nerve regeneration. He showed that if you knock out TET1 or TET2 enzymes, you could not carry out the axon regeneration. And also, there's a very nice study by another team that looked at an ELOF-L2 gene. And the people in the field of cytosine methylation know this gene. ELOF-L2 is one of the most robust biomarkers of human age. If you do an EWOS of age in human blood, you will find ELOF-L2. Now, these people carried out a detailed analysis in mice, and they showed that reversal of ELOF-L2 promoter hypermethylation in vivo leads to increased ELOF-L2 gene expression, and that then rescued age-related decline in visual function in mice. So this was a nice story linking at least one cytosine or one gene in a causal pathway. However, there are plenty of arguments why cytosine methylation is not causal. And one is that out of the millions of age-related changes that we observe, many of these cytosines will have no effect whatsoever on gene expression. That's argument number one. A lot of cytosine methylation is just entropy, no ramifications. But there's also another argument which involves invertebrates. A lot of aging research has been done in Drosophila and C. elegans, and these invertebrates do not have cytosine methylation. So if you make the assumption that aging in worm and Drosophila should be conserved in humans and that the same processes should act, then you naturally reject the role of cytosine methylation. Now, as I said, not all invertebrates have cytosine methylation. However, there are some, and I want to draw your attention to publications by other groups that I liked a lot. There is a clock for European lobsters, okay? So you can measure cytosine methylation in these lobsters and build a clock. And also Tom Little has studied Daphnia magna, and developed a methylation clock for Daphnia. So I just think these are very interesting insights. Now I want to come to humans. In my opinion, the best mortality risk predictor is grim age. And there are really a dozen papers by now by independent groups, in particularly the TILDA team, Cahal, McGrory, but many other studies. So cross-sectional studies show grim age is important. The new frontier is really to carry out longitudinal studies. There are a couple of nice longitudinal studies, but I feel we need more longitudinal studies of epigenetic clocks. I want to draw your attention to a paper from Riccardo Marioni, where that described a large-scale GWAS, over 40,000 people for whom we had both methylation, but also SNP data. And that allowed Daniel McCartney and many other people to build polygenic risk scores for grim age, but also several other epigenetic clocks. And so these, why do I mention it? Because polygenic risk scores can give you another layer or tool for addressing issues of causality. Because that's a big question, causality. Are epigenetic clocks causal, or parts of it are causal? And here I just show you the study from Dr. Kenny and Carl, where they looked at grim age, and they found nice association with several lifestyle factors. So yes, if you exercise, then your grim age is reduced, which implies lower mortality risk. There was also some evidence, weaker evidence for drinking. And of course, a huge effect of smoking, which makes perfect sense to me, because grim age leverages an estimator of smoking pack years, based on cytosine methylation. I want to draw your attention to a couple of other studies that have come out. This was a study that investigated incident atrial fibrillation, studied by Gregory Marcus and Jason Roberts. And here we looked at 5,600 individuals, pretty large scale studies, and 900 of those had incident atrial fibrillation. And sure enough, a 5-year increase in epigenetic age acceleration according to grim age was associated with a 19% increase in the adjusted hazard ratio for AFib. So interesting. I want to come to another age-related condition, clonal hematopoiesis of indeterminate potential. This is known as CHIP, but also known as ARCH. And so CHIP is strongly associated with age, and carriers of these somatic mutations have an increased risk for developing various blood cancers, but also heart disease. And so CHIP originates when hematopoietic stem cells acquire random mutations, typically in epigenetic factors. And I'm aware of 2 publications that have shown that CHIP or ARCH are associated with increased epigenetic age acceleration, Tamir Chandra, Riccardo Marioni. But I will now focus on this study from Siddhartha Jaiswal and Daniel Nachun. And here, Daniel investigated 5,500 people for whom we had methylation data and SNP data. And we could show that all epigenetic clocks showed age acceleration in CHIP. But interestingly, grim age showed the lowest. It's actually the pan tissue clock showed the highest epigenetic age acceleration in CHIP. And that makes sense to me, because to me, the pan tissue clock measures the properties of hematopoietic stem cells. That I always felt that way. But in any event, the interesting thing is that mutations in DNMT3A and TET gave rise to positive age acceleration. But CHIP is also accompanied by mutations in DNA damage response genes. And interestingly, there was no effect. So DNA damage response mutations were not associated with age acceleration. And now you can ask the question, so given that epigenetic age acceleration is associated with CHIP, can't you use that fact to build better predictor of mortality or heart disease? And that's exactly what Daniel did. So he formed different strata. So people who had a CHIP mutation and had positive epigenetic age acceleration, these people, you see the purple curve, showed the highest hazard of mortality and also the highest risk for heart disease. Conversely, people who had a CHIP mutation but did not have epigenetic age acceleration, these are the people in this lower curve, they were protected. So to me, this is really remarkable that epigenetic clocks allow you to stratify the risk of people with a CHIP mutation. And I mention it because to me, this is kind of a prototype study of what I hope we can achieve with epigenetic clocks, which is, can we use epigenetic clocks to inform patient care, for risk stratification? In other words, we need to find ways to develop epigenetic clock applications that translate to the clinic. I now want to come to another topic: in vitro studies, human cell lines. Think fibroblasts, keratinocytes, HUVEC cells. There's a nice paper from Ken Raj, and which addresses the question, which hallmarks of aging relate to epigenetic aging? And Ken Raj did very comprehensive in vitro studies, to address that question. And here's the upshot. When we look at hallmarks of aging, as you remember, there are actually 9 hallmarks of aging, although here I think we only have, I show you fewer. Or is it 8 hallmarks? One, the proteostasis is missing. In any event, there are 4 hallmarks of aging that have been linked to epigenetic clocks: deregulated nutrient sensing, mitochondrial dysfunction, stem cell exhaustion, and altered cell-cell communication. However, other important hallmarks of aging, cellular senescence, telomere attrition, and genomic instability do not seem to relate to epigenetic clocks, at least in vitro. And I will now come to some details. So there are several different ways of inducing inducing cellular senescence. You can do radiation. You can express oncogenes. You can express. Trigger senescence by mitochondrial disruptions, but also, of course, the famous replicative senescence, the Hayflick limit. And so Ken looked at all of these. And the bottom line is, none of these ways of inducing cellular senescence was associated with epigenetic aging in this study. Except you see the red dots, these are the cells from different donors, I should say. Each cell corresponds to a different donor, but you see the red dots lie above the other dots, and so the methylation age was increased in replicative senescent cells. But there's a caveat, right? In order to achieve replicative senescence, these cells had to be passaged or cultured for up to 6 months. And therefore, the hypothesis is that, well, maybe the time needed to achieve replicative senescence really led to this increase. And that's really the case, as I will show in a second. But the other ways of inducing cellular senescence were carried out in 2 weeks, right? Radiation for a couple of days, or RAS oncogene induced senescence. And so there wasn't enough time, so to speak, for the methylation age to increase. And also carried out experiments in telomerase or third immortalized cells. So here the red dots show you age-third immortalized cells, and the x-axis shows you cumulative population doubling levels. And you see that even in these immortalized cells, methylation age continues to increase. So if you prevent replicative senescence, you do not stop epigenetic aging. So bottom line is, epigenetic age is distinct from the best characterized hallmarks of aging, cellular senescence, but also telomere attrition, right? Now, when it comes to genomic instability, Ken carried out very comprehensive studies that were based on irradiation. You can have acute irradiation, 20 grays, or low dose gamma irradiation, 1 milligray, very different pulsings, and also different cell types. You know, you can look at keratinocytes, fibroblasts, you can study mouse embryonics, fibroblasts, different species. In none of these studies did Ken observe that epigenetic age was increased. And so what does that teach us? Double strand breaks and really don't seem to increase epigenetic aging, which is disappointing perhaps, but it just teaches us something about the biology. Now, another hallmark of aging is stem cell exhaustion. And there are links. There are several studies. Here I show you one from Ken that show that adult stem cells sometimes, but not always, can be younger than differentiated cells. So here the stem cells in keratinocytes are actually younger than keratinocytes. There are always exceptions to the rule, and the most obvious exception is hematopoietic stem cells. So if you look at hematopoietic stem cells, they are not younger than blood. Just want to mention it. But long story short, there is a relationship between stem cells and methylation age. Nutrient sensing, another famous hallmark of aging, is strongly linked to the epigenetic clock. And we know that from in vitro studies. If you administer rapamycin to keratinocytes, you slow epigenetic aging. But also there are other studies, caloric restriction in mice greatly lowers epigenetic age in liver samples of mice. There are several publications. But also human epidemiologic studies show it. If you look at even body mass index versus epigenetic aging in human blood, you will see a weak negative correlation. I will have to stop soon. I want to briefly mention mitochondrial disruption is somewhat related to epigenetic aging. You find some applications where you see a strong effect, others where you don't. And let me see. The final hallmark I want to discuss is altered cell-cell communication. In my opinion, there's a strong relationship. So here I show you the results from a preprint. It's on bioRxiv. It's not yet peer-reviewed, where this describes young plasma treatment, a study in rats. So Harold Katcher and Akshay Sanghavi developed a young plasma treatment and applied it to rats. And I think you see this very much rejuvenated the age of the rats. These were old rats, and the treatment, the red bar, the control groups, the blue bars are the treated rats, age-matched controls, and you see drastic epigenetic rejuvenation effect in heart and liver, blood, you know. So, no, sorry, I misspoke. The treated are the orange dots, the orange bars. Sorry, so you need to compare red to orange. And so the treated animals showed strong age reversal in their tissues. So we are now doing replication studies of that phenomenon. So I'll stop with the conclusion. Epigenetic clocks have been linked to 4 hallmarks of aging. Epigenetic age is distinct from telomere length and cellular senescence. Castration slows epigenetic aging in ear samples from sheep, but not in blood. You can use cytosine methylation to build pan-mammalian clocks. You can estimate maximum lifespan and other life history traits in species. We have clocks available for probably all model organisms you can think of. And yeah, here is my acknowledgement slide. I think I mentioned names before, but yeah, thank you. I'll stop with that. Do you have any questions for me?
Just want to begin, Professor Horvath, by thanking you so much on behalf of TILDA, the School of Medicine, and Trinity College Dublin for giving us an absolutely brilliant presentation there. It's incredible, actually, the depth of your knowledge across different fields: genetics, statistics, human species, mammalian species, and so on. So as some of the questions are starting to come through in the chat, the first one I'm going to kick you off with a very simple question. If we stop the clock, will we still get diseases?
I can answer that. The answer is yes, absolutely. And the reason is many diseases have nothing to do with epigenetics, you know. So extreme example, you can get a viral infection and think something terrible, Ebola, right? And a couple of days later, you're dead. And it may have zero effect on the methylome, you know. So unfortunately, we will always die, even if we have the perfectly young epigenome, you know. But I think another question is, well, let's say, how much benefit would you get if you actually kept the epigenome perfectly young, you know? And I talk about the epigenome because I want to be deliberately broad. It's not just cytosine methylation, right? The histone modifications, you know, and some people think of non-coding RNAs as an epigenetic mechanism. So how much benefit do we get from that if we kept our epigenome perfectly young? I think there would be a tremendous benefit, but it's just a hunch, you know. So I think this is really for future generations of researchers, right, that have actually tools to keep the epigenome young, you know. And then they will say, this really led to a jump in health span by X number of years, you know, but I just don't know. Yeah.
Okay, so the questions are flying in faster than I can read them. So what is the role of metformin in EPIC, and any updates in relation to the TAME trial?
Yeah, that's a good question. I'm not 100% plugged in with the TAME trial. You would need to talk to others about it. I know they were certainly planning to measure methylation in the TAME trial. You know, I was very happy to hear that, you know, but I don't have any insights on that. You know.
The next question is from an economist. Fascinating talk. Have you looked at the effects of short-term stress events on epigenetic clocks and economic shocks such as recessions?
Yeah, I personally have not looked at it. I've collaborated with people who have looked at it, you know, and the interesting phenomenon seems to be that short-term stress does not seem to have a strong effect. however, long-term stress, you know. So there is quite some literature, for example, on post-traumatic stress disorder in soldiers. There was a recent study that showed that PTSD was associated with grim age acceleration, you know. And there's also a literature on severe stress induced by child abuse, you know, which is arguably one of the most severe forms of stressing people throughout life, really. This bears with them. And that was also associated with epigenetic age acceleration. Whether short-term stress, such as, I'm just talking about divorce, you know, losing your job, or whether that has an effect on the current epigenetic clocks, I would say probably not. But more broadly, could that affect specific cytosines in the genome? I think absolutely. You know, the human genome has 28 million cytosines, you know, and some of them undoubtedly will be affected by short-term stress. You know? Mm-hmm.
Okay, this is one from one of our external collaborators. Given the motivation to develop second-generation clocks trained on mortality to improve clinical utility, does training pan-mammalian clocks on chronological age limit the potential preclinical utility?
Yes, yes, it does. You know, so absolutely. So the current clocks, the third generation clocks, again, they're trained on age/relative age, but and so, yes, it's a limitation that they have not yet been linked to mortality risk, you know, and believe me that I'm working on that aspect, but I will call it a fourth generation clock. So the fourth generation clocks will be pan-mammalian clocks that actually demonstrably estimate mortality risk in several species. You know, whenever you estimate mortality risk, you need large studies. In humans, we have that, right? The TILDA cohort or others where we have outcomes, you know. But for many animal studies, we don't have that, right? How do I get my animal samples? These are people from zoos, and they don't carry out lifespan studies, you know. Mm-hmm.
Another question from one of our senior PIs here in Trinity College. Again, just echoing thanks for a brilliant talk. What is the reason for skin but not blood effect in castrated sheep?
Yeah, I think I have a good theory about it. You know, it really depends on receptors, you know, for hormone levels. So certain cells, for example, keratinocytes, apparently have a lot of receptors for various hormone levels. And by contrast, blood cells less so. And so I think that explains it, the biology of susceptibility. I want to briefly mention that in the past, we studied the effect of post-menopausal hormone therapy in women. And we found that there was a very significant association in keratinocytes, again, in buccal cells and skin cells. Women who took menopausal hormone therapy, their buccal cells were younger than untreated women. But again, in blood, we saw no effect, you know. So I think blood may not be a good tissue for detecting the effects of hormone treatments in general.
Yeah, there's so many questions coming into the chat. I don't think we're going to be able to get to them all. But one of the questions here from one of our very promising PhD candidates, and I know we've discussed this before as well. What are the effects of medication on these clocks, particularly those that interact with DNA, such as chemotherapies, etc.?
Yeah, if you do a Google search, you will find a couple of papers where people looked at chemotherapies, you know, for example, in breast cancer patients, and also radiation treatment. And there were some interesting results. So women who underwent radiation treatment, interestingly, their blood samples exhibit accelerated aging, which makes perhaps sense because you however, chemotherapy sometimes showed the opposite effect, you know. So be mindful that drugs can have the opposite effect, on epigenetic age. So, but I invite you to read the literature. It was complicated. Yeah. But one reason why I'm working on epigenetic clocks is, of course, to find anti-aging interventions, And there's always the hope that one can repurpose drugs that were developed for cancers, for slowing epigenetic aging, So it's a nice idea.
This question's come in a while now. So are you familiar with Dr. Michael Levin's from Tufts University work on bioelectricity? If so,
Yes.
I have two questions. Flatworm planarians apparently don't age. Any thoughts? And Michael Levin recently regenerated frog limbs and mentioned you sampling the epigenetic age of the regrown limbs. And again, any thoughts you'd have would be appreciated.
Yeah, I've talked to Michael Levin. I think it's absolutely stunning research. I don't have any data on flatworms. I'm glad you asked the question because I'm going to email Michael right after this. But we do have data on African clawed frogs, so apparently it is possible to build an epigenetic clock. I mean, we have a prototype of a clock for African clawed frogs. It was based on, I want to say 48 samples, so I'm still a bit nervous about it. I want to see a replication, but it seems to work, And what is so striking about it is that we actually applied the the mammalian chip, right? The mammalian chip, as indicated by its name, is designed for mammals. But we occasionally have applied it to other vertebrates, as I mentioned, the frog. We also applied it to even fish, because a couple of hundred cytosines also applied to fish, and in frogs, several thousand cytosines. Yeah.
So just to give you a rest, Steve, after a long session, I'll ask two more questions, if that's okay. One of them I think is a really great one, and we've recently received telomere data in the TILDA study, so I'm interested in this too. Once again, thanks for a really great talk. I just wanted to explore the telomere length link with epigenetic changes. I'm wondering about your thoughts of how epigenetic changes in telomere-related genes could affect factors beyond telomere length.
Yeah, I mean, I think it's a very important question. I'm very happy that you will work on it, So, and I'm happy to help you if you have, if you're interested. Not that you need me because you know all of my tricks and way more than what I know. But anyways, it's complicated, So, when we did genetic studies, GWAS, of age acceleration, we did find evidence of several telomere-related genes, So that's fact number one. And also, we published actually a large EWAS study, right, where we correlated cytosines to telomere length to find which cytosines correlate, And all of these studies show that there is some sort of a connection between telomere maintenance, and methylation changes, And so, yeah, there is evidence, there is a link. But it is complicated. On a superficial level, there is no connection, right? I showed you these results from these in vitro experiments that decouple it, right? This third immortalization. I also want to briefly mention, you can, of course, correlate leukocyte telomere length with epigenetic age and remove the chronologic age effect, right? And you will see very weak correlations, So on a superficial level, there's no connection, but a more detailed analysis shows there's one. Sorry, I'm vague, but yeah, just you guys will have to carry the torch on that probably.
And a very final question for you, Steve. Again, incredible talk. What is your hypothesis as to why cellular senescence, telomere attrition, and genomic instability are not linked to epigenetic aging? Shouldn't methylation have an impact on these cellular mechanisms?
Yeah, I mean, it should, you know, I need to say, I always show you the results from some experiments, and biology is always so complicated, you know, because I could have shown you evidence that actually links genomic instability to epigenetic aging. So for example, David Sinclair has some nice mouse models that link it, you know. Yeah, so why is there no connection? I do think aging is really multifaceted. You know, there are really different drivers of biologic age. I would love it if we could reduce it to one main driver. You know, as a former physicist, you always want the grand unified theory, you know, where you explain everything, but biology seems to be messy, you know, so it could be there are different ways of leading to organ dysfunction, you know, they may or may not be coupled, you know. Mm-hmm.
Yeah, that's actually a sentiment that we've echoed even within our own TILDA team about whether aging is dimensional or multidimensional. And Steve, with that, I'll say once again, thank you very much on my behalf. I think I'll pass you back now to Professor Kenny, who might just want to close the session.
Yes, thank you so much, Carl. Yeah.
I do want to close it by saying thank you so much. That was an incredible talk. Thank you.
Oh, thanks.
Lots of food for thought, and we will be in contact, of course, in the near future with respect to some of the issues you've raised and some of the potential for TILDA in collaboration. So thank you so much on behalf of Trinity College, the School of Medicine in Trinity, and the TILDA research and aging research teams, Steve.
Yeah, thank you. Goodbye,
Much
everyone.
appreciated.
It was
Great
a real
talk.
pleasure. Bye-bye.
Thank you.
Thank you.
Thank you.